Okay, guys, let's get started. Oh, you guys are excited today. Okay. So today we are going to start our third unit on the cytoskeleton. Well, it's not our third unit on the cytoskeleton. It's our third unit, comma, on the cytoskeleton. So, quick business. Exam 2 will come back on Monday. I promise, kind of. So far, so good. Lots of over 100s and lots of not so over 100s. I don't know what to say about that. It's all of you. Okay. Thursday recitation, whoever's in Thursday's group, please remember to print out the You Be the Scientist file. That is an exercise where you use data. Don't mind me. Where you use data to basically go through the experiments yourself, and you are actually analyzing the data and making hypotheses based on the data that's given to you. But without this piece of paper, that's very hard to do. Okay, so let's start out. What is the cytoskeleton? You guys all took 201. Kind of. Hopefully. So what is the cytoskeleton? Yeah.
It gives a cell like structure and support internally.
Okay, structure and support. What else? Transportation. What do you mean by transportation? Motor, vesicle things. I like it. Okay. Yeah.
Technical terms.
Technically, yes. What else? How about this side of the room? Cytoskeleton. We've got structure, we've got motor, vesicle things.
Composed of microtubules.
Composed, where is that coming from? Oh, composed of microtubules, thank you. Yeah, microtubules. What else makes up the cytoskeleton? Microfilaments, intermediate filaments. Yeah. Actin, right? So you guys have seen, 3/4 of you have seen, hopefully, I don't know if we have equal representation in this room right now, have seen already what E. coli does to encourage pedestal growth by kind of hijacking the active polymerization system in recitation. Okay, so we've got what it's made of, we've got what it does. There's some things. What's a major process that is controlled? Okay, you didn't even heard mitosis. Who saw my lecture already? Oh, I forgot to post my lecture for you. I'm sorry. I'll post it right after. Yeah, mitosis is controlled by what? Okay, microtubules. Microtubules make up the mitotic spindle, right? Okay, so what I have here, I love immunofluorescence, as you guys may have noticed already. So this is a great opportunity because actin and tubulin are great for pretty pictures. So there's a bug there, and it hit me in the head before. So if it hits me in the head again, just call 911. So here you've got blue. Tone it. It's gonna be okay. Here you've got blue. Blue is DAPI, which stains the DNA. Red is actin. And green are microtubules. So how many cells do you think we have here? 9. Okay, good. So you guys are getting good at orienting yourself. Okay, so this is what. Now, I haven't taught you anything yet, but you guys did some reading this weekend to do your quiz. What phase of the cell cycle do you think these cells are in? No, they're not splitting, but so I want you to pay attention to your microtubules. Interphase, right. Okay, and we'll look at some more pictures. Okay, so that's good. Good start. Okay, so I want to start with some inspiration, yes?
Why is it on the interface?
Why, the question I just got was, why is it interphase? What's the difference between microtubules during interphase and during mitosis? Hmm? To be created, give me more than that. Well, you know, we're gonna get into, yep.
Because they're not like, I don't know, really gonna say, but they're not like running the expansive cell, pulling it that way.
Ah, okay, so they're not
It's there.
forming the mitotic spindle
Yeah,
where
they're
they're,
not
yeah.
forming the thing.
Yeah, so you'll see, we're gonna see some pictures shortly, and you'll see that the microtubules reorganize at different phases of the cell cycle. Okay, but I wanted to start with some inspiration, because when you start learning something new, it's good to care about what you're learning about. So there's a few videos in here, some pictures, and we'll see. So here's mitosis, and so again, here you've got blue for your DNA, red for mitochondria, but green for microtubules. And you'll see in interphase, there's just this network of microtubules running through the whole cell. But what happens over here, starting at prophase? You see, you start to get your duplication. You actually start to get formation of your mitotic spindle. So you're going from all of your microtubules coming out of your microtubule organizing complex, MTOC. And then it duplicates, your centrosome duplicates and becomes your mitotic spindle. And so that's what you see happening here in prophase. And then you see attachment to, so remember DNA is blue. You see attachment to your chromosomes. And here in anaphase, you're starting to see chromosome segregation. And then telophase, cytokinesis. And if you saw, if the red was actin, it would be even cooler. And I think I have that in a little bit. Because then you would start to see actin right here and right here specifically, because that's your contractile ring that actually separates your cells. Cool, huh? Yeah. Okay, so that was mitosis. So then there's this concept. So there's a lot of concepts involved in studying cytoskeleton and cytoskeletal proteins that are important to understand. So here's one that you guys really need to understand: dynamic instability, because that's kind of what it's all about. And dynamic instability is the fact that you've got, especially when you're talking about actin and tubulin and microtubules, you'll have this balance between monomer, so tubulin dimers, and polymer microtubules, and actin monomer and actin filaments. And so there's this balance, and there's this constant growth and disassembly of your polymers. And so that's dynamic instability. It's the fact that there's this constant growth and disassembly. So this picture is showing a time lapse of GFP labeled microtubules. So each one is a different time, and you'll see that there's the length of these microtubules is changing. And I have a movie that will show it even better. So you're going to have to, yes?
Oh, is it, you said shifting from actin monomer to actin filament?
Yeah, so think monomer to polymer.
Okay.
Yeah.
Just to make sure I heard.
Okay, so this has some audio with it, but because I'm using my computer, I haven't quite figured out how to make the audio come through, but that is okay because I can tell you what's going on here. So these are microtubules. So they actually put a centrosome, so that's where your microtubules are coming from.
And then shrink back
Above.
rapidly.
They put centrosomes and microtubules in culture, tubulin subunits, and you can see that they're growing and shrinking. And each line is one microtubule coming out of the centrosome. Should I play it again? Would you like that? I see you, Tyler. Let that go once more, maybe.
Microtubules continually grow from the centrosome
So
added to
this
a
is
cell
in culture,
extract.
not in the cell, which is why you don't see anything.
Quite suddenly, however, some microtubules
You can
stop
do this in
growing,
culture.
and then shrink back rapidly,
Okay.
a behavior called dynamic instability.
Right, so you're referring to how you need to get the aggregation of monomer to start polymerizing. Is that what you're talking about? Right. So for microtubules, it starts at the centrosome because it's made of it's the microtubule organizing complex, and there's a ton of gamma tubulin ring complexes there, which act to nucleate and form that first stable aggregate where your microtubules will grow from.
So nucleation is kind of like simultaneous with there's a nucleation in microtubule.
Yeah, so you need nucleation for growth. And so we're going to get into this as we start going through this. Okay, any other questions? Isn't that cool? Okay, another cool thing to inspire you to love the cytoskeleton. So organelles and vesicles. Remember we talked all about vesicular transport for about 3 weeks, right? So vesicles and organelles move around in the cell, and they actually walk not really walking, but they're bound to motor proteins that walk along these different cytoskeletal structures, either actin or microtubules. And so I have another movie here for you. And so this is going to take us. Okay, so this is showing mitochondria. Let's see if I can. Okay, so that one is rabies virus infected, so that actually rabies virus inhibits the movement. But let's replay it once more. So this is an uninfected neuron. Do you see them moving around? Now clearly that's sped up a little bit. And those are mitochondria, and they're attached to motor proteins, and they are walking along the cytoskeletal proteins. Once more for fun. Isn't that cool? It's kind of amazing that you can take these movies. And then when you... they're very sad mitochondria. With, oh my God, I love it when you connect things from different... Okay, let me get out of here. The question was, does SRP use the cytoskeleton? What do you guys think? Let's go back to this. Okay, does everyone remember SRP? What does SRP do? Okay, you're not allowed to forget things in this class. Yeah. So it attaches to the signal sequence, and it takes the whole complex to the ER, right, to translate proteins destined for the secretory pathway into the ER. And so the question was, does SRP move around like this? And so I would say, so the key is, does it interact with a motor protein? And so I would say SRP itself, probably not. But I'm not sure, and we can make this a stump the teacher if you'd like, but let's make it one step forward. What happens after when you make your protein in the ER and then you bud out into a vesicle, those vesicles are moving this way because they're attached to motor proteins and moving along microtubule tracks.
To go to the Golgi.
To go to the Golgi, and then onwards to wherever else they're going to go. Should we make the stump the teacher question? So we should explain to our guests in the back, who I should be ignoring, that whenever my students ask me a question that I did not answer, I offer them 1 point if they can get the answer to me within 24 hours. Referenced by email, not copied and pasted, but actually written. So the question was, does SRP use motor proteins and walk along cytoskeletal structures to get to the ER? Does SRP use motor proteins to bring SRP to the ER? Okay? Yes.
I don't know if this is what we're about to talk about, but I thought everything you use in motor proteins. That's not.
So you thought everything used motor proteins?
It
Maybe.
just seemed like that's the function they do, so
Maybe. I think there's a lot of just movement. When you talk about proteins moving within the cell, a lot of the time there's just a high concentration
Okay.
of things that are then going to bind to each other just by movement. Just by like thermal energy, movement via thermal energy. But larger structures like vesicles and organelles are definitely moving around this way. Okay. Next. Okay, so this is going to show organelles moving on microtubule tracks using organelle-bound motor proteins.
In this experiment, a cell homogenates
And so you
between
can
many
actually
different
see there's
organelles
your
is added to
microtubule and there's your
microtubules.
organelle.
Motor proteins are normally attached to the organelles. When ATP is added as a fuel for
And
the
so they
microtubules,
add
some
ATP
organelles
and they're
bind
showing
microtubules
movement
and are moved along the tracks
of
by
these
their motors.
Organelles
Most
along
kinesin
these
motors
tracks.
move towards the plus end of microtubules.
And so they're gonna learn as we start to learn more about
Dynein motors always move in the opposite direction.
microtubules and actin and the
Both
types
motors
of
are used
motor
to transport
proteins that
organelles.
work on them.
And occasionally a single organelle.
They are
which
directional.
must have
Some
both
move to the
types of
plus
motor
end and some
attached,
move to the
can be
minus
seen
end,
to
and so
switch
that's what they're
directions.
showing here.
The bidirectional traffic observed here is reminiscent of that in an intact cell.
Okay. There's a really cool one coming up that I was so excited to show you. I think this is it. Okay, so this is showing vesicle transport also bind to motor proteins, walk along microtubules, and here you're seeing vesicles, and they've got their head domain and they've got their motor domain, and their head domain tends to bind to the vesicle or whatever is being moved, and you can see you've got a dynein and you've got kinesin, and they're directional. They walk in specific directions and are used for different purposes. Yes.
Diamonds used to pinch in endocytosis.
That is dynamine. Dynamine. So you're confusing the dynamine, which does that when you're talking about endocytosis and pinching off of vesicles. That's dynamine. And this is dynein. I know, they sound the same. The scientists are all yeah. Okay, so this is a very, very cool movie. And I promise you I'll stop entertaining you with movies in a minute. Okay. So this one I'm going to move forward a little bit because it's really long. So I'm just going to, so this is in Arabidopsis, but this is the cool part. So they're showing it from like the seeds germinating to making the actual plant, but then they're going to zoom in. And so you can see all these little vesicles moving around in the root tip. And it's even cooler. And so you're seeing the vesicles moving, but now you can start to see them attached to the motor proteins walking along. So there really is showing. And so this one is clearly actin. This is not microtubule. But isn't that cool? And so that's a vesicle. So that's a vesicle containing protein. So cool, right? Okay, and that's the cool scientist. Okay. Stop. Okay. Okay. Am I getting you to care about the cytoskeleton? Okay, good. Okay, more for. So we talked about movement of organelles, we talked about mitosis. This is going to show how actin is involved in movement. Someone said movement when I was asking what the cytoskeleton was involved in, right? And so this is going to show a neutrophil chasing Staph aureus. So this one, right, these big red ones are red blood cells, and this big blob is a neutrophil, which is a white blood cell. And then you can see right over here, there's these little dots. Those are your Staph aureus, and it's going to actually chase it around, and that's controlled by actin polymerization, which I want you to start thinking about the paper we did in recitation and how there was recruitment. Sorry for those who haven't done it yet, but now you'll have a leg up tomorrow. There was recruitment of ARP and WASP, these nucleating proteins, to the site where actin needed to be polymerized, and that's how it works. In this case, E. coli was kind of forcing it to happen, but it's similar with neutrophils. Okay.
Neutrophils are white blood cells that hunt and kill bacteria.
So in a
In
minute,
this spread, a neutrophil
this
is
guy's
seen
gonna
amidst
start
red blood
chasing.
cells.
yes.
Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is
Ricin?
sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to
Let me
avoid
try
their
to think
predator.
about that.
Eventually, the
Look
neutrophil
at that.
catches up
See,
with
so
the
it's
bacteria.
chasing,
and
and
engulfs
so you
them
can
by
see it's got this
phagocytosis.
leading edge right over here, and so they are phagocytosis. So got it. Neutrophil wins. And actually, this is all going to be online, so you can listen to it. It actually burps in the end, which is kind of funny. Okay, so Tyler was just asking about the paper we did in recitation about ricin toxin, and you were making a connection. Oh, the toxin was going through the Golgi to the ER. That's how it works, inhibit translation. And they were actually identifying drugs that inhibit that. So,
That's pretty much what they were inhibiting the connection.
were they? Oh, we have to go back and look at that. Let's discuss that later. Okay. So cytoskeleton. So again, we've got our, so this is showing blue DAPI, DNA, green microtubules, and here the actin is stained in red, and you can see it along the edge of the cell. Okay, so what role does the cytoskeleton play in complex cellular processes? Well, if you have to think about it, cells, we already know they need to interact with their environment. They change their shape, move from place to place. They grow, divide, respond to extracellular signals. They pull chromosomes apart during mitosis. There's intracellular traffic. Sperm need to swim, otherwise our species is not going to do very well. White blood cells crawl across surfaces. And of course, muscles contract. So you could probably connect the cytoskeleton to a lot of things we've spoken about already in class. And then a lot of things we will speak about in our last unit about cell cycle. Okay, so the key players of, so now everyone cares, right? Are you guys all committed to the cytoskeleton? No? Okay, well. Here are the key players to the cytoskeleton. You've got your intermediate filaments, and so they're important for mechanical strength, resistance to shear stress. Microtubules, they are involved with positioning of organelles. They direct intracellular traffic. Obviously play a role in forming the mitotic spindle. Actin filaments, they're involved with the shape of the cell and the movement of the cell. And then there are the accessory proteins. So there are many proteins that we're going to go through, and their role is to either link cytoskeletal proteins to organelles or to each other, or control any of these processes, usually through regulating whether or not you're going to get assembly or disassembly of your actin filament or microtubule. Okay? And so when we were speaking about WASP and ARP, those are two of these accessory proteins that are involved in encouraging actin polymerization. Oh, and then in the paper also there was alpha-actinin, which was a cross-linking protein. Okay, so this is just an example, right? So the cytoskeleton is very dynamic, it's constantly changing, and it needs to be changing because the cell needs to adapt to its environment. So this is just an example of different changes that occur in the cytoskeleton that's associated with cell division, right? So here you just have your normal interphase fibroblast, and it crawls, it moves, so it's polarized, and it has a dynamic actin cytoskeleton. So the actin is going to move to in the direction that the cell needs to move in. So here you can see your leading edge. Here you're going into mitosis, and you're going to get your microtubules are going to reorganize. You can form your mitotic spindle. And then you're going to align and segregate your chromosomes, so that's dependent upon your microtubules. And then your contractile ring is going to form to pinch off into two daughter cells during cytokinesis. There you go. So if there's anything you learn, is that these cytoskeletal filaments are dynamic. They're constantly changing in response to stimuli. Microtubules reorganize at different points during the cell cycle, and reorganization is constantly occurring through growth and disassembly of these filaments. Okay? Any questions so far? Okay. So at interphase, microtubules are organized at the nucleus. So they're coming right out of one central location. And then they're going to reorganize to form the mitotic spindle at cell division. So you've got your centrosome duplication, and then you form the mitotic spindle, and that's going to bind to your chromosomes, line them up, and then through disassembly of your microtubules, segregate your chromosomes into what will become the two daughter cells. And so here it's a little different. Your green is still your microtubules, and here the chromosomes are labeled in red. And they've chosen to label the peroxisome blue. So these cytoskeletal filaments are constructed from smaller subunits, and then they reassemble in response to external signals. So remember, generally, things aren't random in the cell. Well, there are things that are random in the cell. But usually there's signals. So we talked a lot about signaling in the last unit, about signaling in terms of activating gene expression from a receptor, like a ligand binds to a receptor, and then there's a signaling cascade, and then you get a transcription factor that goes to the nucleus and activates gene expression. Well, also you can get signaling that's going to activate actin polymerization in a certain region of the cell to get movement, for example. So these filaments are made of small subunits that are going to assemble to form these large polymers. Okay? So in this case, in this example up here, you might have all your actin at this end, all your actin polymers at this end, but maybe there'll be some kind of signal, such as a nutrient source, and then you'll get this rapid disassembly of your polymer and reassembly at the new site in order to change the direction of the cell. So you can imagine in that neutrophil movie we saw, that's what was happening. There was something being secreted by the bacteria that was saying, hey, I'm here, and the cell said, okay, disassemble actin, run across the other side of the cell, reassemble actin, let's run this way. And had to keep doing that because it was going in many different directions. So in the book it says it's like building a skyscraper out of bricks, because you can think your actin polymer is made of many individual bricks. Yeah.
Does actin like walk down actin to get to the other end of the cell? Like what moves it gets the walkway?
So the question is, how does the actin get to where it needs to go, right?
Because it's like the pathway, so it can't walk on its own.
So actin's not walking down actin, but that is an interesting way to think about it. But you think there's all these soluble subunits, and they have a high concentration, and they're recruited by different proteins. And we're gonna learn about all of these proteins that are involved in encouraging actin polymerization. Yes?
So would it be like, there was a first question about on and off and, right? So would it be like, where there's hydrolysis and where there's not, it's gonna move along that pathway?
Okay, so the question was referring to the quiz question about the critical concentration, right? And about, so you're thinking about treadmilling and how, okay, so we're gonna get into this, and how your on and off rates and how that's gonna cause treadmilling. No. And we're gonna, as we get more into talking about this, it really has to do with recruitment of actin monomers to where they're needed. And so that's kind of what happened in this week's replication paper, where E. coli injected a protein into the host cell that was phosphorylated and recruited a protein that then recruited ARP, and ARP is a nucleating protein, and that then recruited actin subunits to polymerize actin at that location. But there's such a high and it all has to do with the concentration of actin monomer in the cell. So there's a high concentration, and so if it's busy in a polymer at this end, and then it disassembles because some proteins are mobilized to encourage disassembly, then there's going to be a high concentration of monomer that's available to be recruited to this end of the cell. And we'll talk more about the signaling that kind of controls that. So good question. Any other questions? Okay. So these filaments are formed from multiple protofilaments. So that means that you get these polymers forming from individual subunits. And so this allows for strength and ease of assembly and disassembly. Because imagine if you didn't have this ability to assemble where you're needed and then disassemble and then go somewhere else. Imagine like dragging around a huge microtubule through the cell. I don't know, seems like it's not as effective. So intermediate filaments. I don't talk too much about this in this unit, but so they're another cytoskeletal structure, and they're important for strength, tensile strength, and bending. So remember, we don't have a cell wall. Do we have a cell wall?
No, we're playing.
Do we have a cell wall? No? Okay. Do we have an exoskeleton? Okay, so like we need to be able, our cells need to be able to handle stretching and pressure, and so we need strength, and our cells need to form into these organs that can handle different pressures, and that a lot of that comes from the properties of intermediate filaments. And so these are made of these subunits that, here, let's go here. So there are these monomers that are assembled into dimers and form tetramers, and then assemble into these sheets that supercoil into this rope-like filament, which to me reminds me kind of like a cable, and has that kind of strength. And that's what gives our cells and our tissues the strength that they have. And so these intermediate filaments are required for formation of tissues and organs, because the cells need to be connected to each other and stay connected to each other. and to the extracellular matrix to form these sheets. Okay, so let's talk a little bit about the language of this cytoskeleton. Okay? You have to learn a certain number of vocabulary words to really understand it. So the first one, I'm just going to give it to you as a list, and then we'll talk a little bit more about it. So first one is nucleation, right? So who knows what nucleation? Obviously, I have it right here, but you guys did a little bit of reading over the weekend in preparation for this. What is nucleation? Who understands that concept? I have my random class list, but then we'll find out who's not here. Yes.
And then, when you take the monomers and you start putting them together to the dimerize and then trimerize, and then it forms like nucleus big enough to start polymerizing.
Okay, yes. So you get the monomers that are going to bind to each other, and but they don't really start polymerizing into a microtubule or into an actin filament until you get this stable aggregate, the stable nucleus. Right. And so that's this stable starting unit that allows for rapid polymer growth. And so we'll see a lot of these proteins we're going to learn about are involved in nucleation, which is creating this stable starting unit, and that promotes microtubule growth or actin polymerization. Critical concentration. So this one I got a few emails and questions about over the weekend. Who feels like they understand the concept of critical concentration? Yes?
Is that dealing with like how many monomers are around as to how quickly or fast they build the critical part?
So it has to do with, right, so what Ashley said was it has to do with how many monomers are around and how that leads to how rapidly filaments are going to either polymerize or disassemble. And so I think I have, yeah, okay, so let's just go here for a second. So critical concentration has to do with the concentration where there's monomers in solution, and if your critical, now I'm going to get turned around because I'm explaining it to you guys. The critical concentration when it is it's your fault. So there's this relationship between how much monomers in solution, and so when you're at a certain concentration, you're going to promote growth. If you're above a critical concentration, you're going to add to your polymer, and you're going to have growth of your microtubule or of your actin polymer. And when you are below the critical concentration, you're going to have disassembly of your polymer. And so that ties into the whole concept of dynamic instability because it's controlled by the critical concentration. Okay? Yeah.
So is that just like a threshold that it's one for one way and one for the other?
Yep, so you can think of it as a threat. I think that's a nice way to think of it. It's kind of like a threshold, and when you hit that concentration, so at the critical concentration, you're at this steady state where your adding and your leaving is equal. Yes.
It's just like an equilibrium.
Yes, like an equilibrium constant. But so what we're going to learn about is that all of these different proteins, these accessory proteins that we're going to learn about, are going to mess with that a little bit because you need to force, sometimes to respond to a certain stimulus, you need to force polymer growth or force disassembly. For example, when it's time to enter, go from metaphase to anaphase, you need to force microtubule disassembly. And so if we're just relying on the critical concentration to control whether or not things are going to grow or disassemble, things are never going to happen how we want them to. So there are all these proteins that encourage disassembly or encourage assembly when they're needed. Okay? So that's the concept of critical concentration. Okay. Oh, I have a video for nucleation. because this often confuses people. Okay, so there are your actin, this is actin, your actin subunits, and so when you get this stable nucleus, then you can start to have actin polymerization. And so you notice you have your plus end and your minus end. Things are more apt to add at the plus end than at the minus end, and we'll go over that more. So when you're at steady state, which is at your critical concentration, there's equal addition and removal of your subunits. And so this is your critical concentration. So you notice you're not actually having any growth or shrinkage per se. It's staying at the same size. And so if you're below your critical concentration, then you're going to encourage disassembly. Okay. How are we doing? Everyone having fun? I feel like it's Monday. I think it's because we had an exam. It's actually Wednesday. Tomorrow's Halloween. Okay. I needed a little brain break. Okay. Okay, so let's continue with the language of the cytoskeleton. Okay, so I mentioned something about plus and minus ends, right? Right. So actin and tubulin polymers have two ends at which monomers can leave and add. And this is going to bring up another quiz question in a minute. And they leave and add at different rates. And plus end usually means this is the end where you're more likely to have addition, and minus end is where you're more likely to have leaving of monomers. So I had a few questions over the weekend. Yes?
Yeah, because that question was, when you read, it said like they added both ends.
Right. Okay, so I had a question on the quiz about microtubules, and I said, well, something like true or false, monomer is more likely to add at the plus end, or something like
No,
that.
it just said, in the tubular microtubules add on the plus end.
Yeah. So I had a few people email me, and they were like, no, but it can add at the minus end, but more slowly. But I wanted you guys to look in the context of the cell. Your minus end is the microtubule organizing complex, so there's no way to add any more there because they're all growing out of there. So in reality, you're adding. So once you, because that's full of these gamma tubulin ring complexes, which are nucleating your microtubules. So in reality, with your microtubules, you're adding at the plus end. So in solution in a tube, maybe, but for actin, absolutely. You're adding at the plus end and at the minus end, but you're just adding more rapidly at the plus end, and you're leaving more rapidly at the minus end. Okay? So you guys can take that up with me if you're not happy. Okay, so then there's treadmilling. I think I have a movie for that too. So treadmilling is when, so remember your critical concentration is specific to each end of your polymer. So you'll have addition and leaving at your plus end and at your minus end. And so when your critical concentration of your minus end is greater than the critical concentration at your plus end, then you get treadmilling. And that's because you are adding faster at your minus end and leaving at your plus end, and it makes it look like the whole polymer is moving. questions about this stuff? Yes.
What would be the significance of that? Moving.
What's the significance of treadmilling? So we are going to start to learn about that when we start learning about the cell moving and the cell moving forward. Thank you for bringing that back to big picture. Sometimes we get so bogged down with the details and need to be brought back to the big picture. So when you start talking about the cell moving forward and you're getting this actin polymerization, that's exactly what's happening, is you're getting growth at the plus end and leaving at the minus end, and that's causing that forward movement because you're pushing against the plasma membrane or the cell cortex, and that's moving the cell forward. And we'll learn about that. Yes?
So with treadmilling being involving like critical concentration, you're saying at the minus end, I guess I'm trying to understand how it's critical concentration is delivered on to some extent. So it just means again, minus end is like a larger equilibrium, like more on, more off.
Right.
Larger than the other. It's like,
So that's when so I like that, more on, more off at the minus end. So that means at the minus end, you're getting more adding, you're getting more addition, and at the plus end, you're getting more leaving. And so you can imagine how you're having more addition. You're going this way, and this is your plus end, and this is your minus end. You're having more addition at the minus end, but more leaving at the plus. You're going to kind of move that way. Does that make sense?
That sounded bad.
Did I get it backwards?
Yeah.
Okay.
It's gonna be minus and leaving.
Oh dear.
And then
Okay, so let me see. I think I have a treadmilling video. No? Okay. Let's reevaluate this, okay? We'll get back to it. I promise you. Treadmilling. Oh, now you're gonna get me confused. Yes. Can I draw it? Okay, let's see. Okay, critical concentration. Oh. Okay. So, we'll just make a few. Okay, so your critical concentration at your minus end is greater, which means you're going to be adding more here. Your critical concentration at your plus end is lower, so these are going to be going away.
Moving towards the next left, right?
also maybe you have stuff in
So that means then these will go away, but then these will add. Oh, so you're moving to the left, right? So you guys, you got me turned around. I'm gonna have to go back to the drawing board on this and come back to you guys because I don't want to mislead you. Anyone want to add anything? Yes.
well, I mean, not that I want to keep hammering this around, because you're adding more. I understand what direction it moves in. But I guess my question is, because you're adding and taking away from both sides, are you not taking away?
No.
from
So
both sides?
yeah. So
Not as much.
So exactly. So when you're above your critical concentration, you're more apt to add, and when you're below your critical concentration, you're more apt to lose. But it doesn't mean you're not losing. It's just you're adding at a higher rate if you're above your critical concentration.
So in treadmilling,
Maybe Dr. Wimmer. No.
You really want me to?
No, it's okay. Okay, let's get back. I want to reevaluate this because I'm not explaining it well,
that's fine.
and I don't want to mislead anyone. Okay, yes.
Is that same as the concentration of the minus inside? That,
Yes.
oh, now I get it.
Yeah? Okay, that's good. Okay. We will get back to this. Okay, so we're back to dynamic instability. So microtubules can grow and rapidly shrink, right? And so they're alternating between these two states of growth and disassembly. And this is dependent on the GTP cap. And I'm going to refer to the GTP cap over and over again. And it's best to understand it. And what it really means is it's the GTP-bound form of your alpha-beta tubulin, okay? And it doesn't mean there's a cap of GTP. The students always give that back to me on exam. It's actually when you add your tubulin subunits, your growing microtubule, you end up getting GTP hydrolysis, but it takes a little while. And so you get this buildup of a GTP cap. And I'll show you as we start to look at some figures of the actual alpha beta tubulin. But when you have your GTP cap, so GTP bound at the end of your microtubule, you're growing and you're stable. But when you lose that, and this has to do with the critical concentration, if your critical concentration is very low and you start to have more disassembly, you can't add anymore, you lose your GTP cap, then you get shrinkage of your microtubule. And that will be called catastrophe. Yes?
Just from like, going from GTP to GDP, you can hydrolyze, or you can use like GAP or GAP to just knock off the whole GTP and put in GDP. So there's really two ways to go from like GTP that we really learned about.
Okay, I'm trying to tell you. So you can go, so you can actually have a GTPase domain within your protein, which will hydrolyze your GTP to your GDP. And then sometimes there'll be a secondary protein, which we call a GAP, which actually hydrolyzes the GTP. Okay, keep going.
It actually hydrolyzes it, or does it just like, I don't know, I feel like, or does it just take it off?
It hydrolyzes your GTP to GDP. When we talk about kicking off your GDP, so when you want to add new GTP, then you remove your GDP and add your GTP back, and that's usually either through often through the activity of a GAP.
But it's always hydrolyzed.
So when you're going from GTP to GDP, it's always hydrolyzed. Okay. It's like my oral exam up here today. Okay. No, you guys are doing great. I love it. You gotta keep me on my toes. Okay. So this is just going back. So this is a figure explaining nucleation. So nucleation is that initial step of making that aggregate that allows for actin filament growth, or in this case it's actin, or it could be microtubule growth. And so does this look like a growth curve to you? You took med micro. Yeah, it kind of does look like that, right? So you'll have these actin subunits, and they'll kind of bind, unbind, and eventually they'll form this oligomer, and then you get that exponential growth. But until they form that oligomer, which is actually the nucleus, so it's not the nucleus where your DNA is. It's just a stable aggregate of monomers, which form a stable oligomer. Then you'll get this rapid actin filament growth, or microtubule growth, until you reach your steady state. And so this is your critical concentration, where you've got this. You're at the concentration in the cell where the concentration of monomers is such that you've got equal addition and removal of monomers. So you stay the same size. Okay? Not you. The filament. Okay? Are we good? Yes.
Does the nucleus ever come off?
Does the nucleus ever come off?
Yeah, if you're doing that and it comes from the one end and goes to the other end, like minus one end.
Oh, I see what you're saying.
Does the nucleus ever come off?
Yep, so don't think of the nucleus as a thing. So the question was, does the nucleus ever come off? Okay? And so don't think of it, yeah, it's a peanut gallery. So don't think of the nucleus as something that's like has that function, and it retains that function all the way throughout the whole process of polymerization. Think of it as just like this starting unit that takes off in polymerization, and then it doesn't matter because once you have growth, you're good. You know what I'm saying? So it's not like the GTP cap.
It's not constant.
No. Does that make sense? Okay. Any other questions? Yes. Huh? I'm confused. You have to get loud. Here, you can get to talk louder.
Like what she said, nucleus shifting. So those four, they stay the nucleus.
Yeah, no, so don't think of a nucleus as like it's the nucleus. It stays the nucleus. The nucleus is the action. It's the nucleation is the action of forming a stable aggregate that allows for polymer growth. And so you can call it the nucleus, but it's not like there's a nucleus and then you have your polymerization and then it falls off. And no, it's just like the starting point. Just the starting point, that aggregation that makes it stable and allows to grow. Okay? Yes.
Let me just be clear. Nucleation is not forming nucleus. It's an action that's going to form little blobs or stable things that is going to actually get together and grow.
Yes, yes. So I think you're right. Yes. So nucleation is the formation of a stable clump of actin monomer that encourages actin polymerization and growth into the actin filament. And that concept is true also for tubulin. Good?
Yeah.
You don't look
No.
I mean, you look great.
No. She was like so confused. I was like, it's like Destiny's Child. The first four members just started the group and they just kept changing them out.
Well, you're talking about Destiny's Child. Okay. Pop culture. Gone. Okay. Okay, let's keep going. So let's talk about, so if you want to encourage, we're having too much fun today. Are we having too much fun? How about this side of the room? Yeah. Serious faces. Okay, so if you want to encourage actin polymerization, would it be helpful to just pop in a nucleus, like a seed?
Can you do that? Like the pre-made one?
Yeah. Yeah, because there are these nucleating proteins, and their function is to create a stable nucleus, and that's ARP, for those who read. So we're talking about E. coli recruiting ARP and WASP and actively polymerizing actin. Well, so that's what it does. So there are these nucleating proteins that bypass the nucleation step. Yes?
Is that kind of like when you make a rock candy, you throw something in there to make the supersaturated solution, so it'd be like supersaturation of the
Like making rock candy. I haven't made rock candy.
What?
I know. I know. I'm a
Now you have something to
player.
do with your kids.
I know.
Is that
I think you might be right. I think you're right. Yes, go ahead.
is that what WASP is? The protein that recruits.
So ARP and WASP work together and recruit actin monomers and encourage rapid actin polymerization. Yes, that's exactly what that paper was to drive home. Yeah. Okay, good. Are we happy? Okay. Okay, so microtubules form from tubulin subunits made of alpha and beta tubulin heterodimers. And this is how they look. And so beta tubulin has GTP. Well, so they both have GTP bound, but beta tubulin has the GTPase domain and hydrolyzes the GTP. And so when you're GTP bound, you're more apt to bind, and when you're GDP bound, you're more apt to leave. And so that's involved with this kind of dynamic instability. So when we talk about the GTP cap, that's when you have a whole lot of GTP bound dimers, but when your critical concentration gets low and you start to leave and you can't add anymore, you lose your GTP GTP cap and you start to disassemble. Okay? No gaps. Okay, so a microtubule is a hollow cylindrical structure, and it is built from 13 parallel protofilaments. So we just looked at one. So this is this alpha beta binding of your tubulin dimers. Well, they're going to line up and form this tube made from 13 parallel protofilaments that are made from alternating alpha beta tubulin subunits. And so you've got these multiple contacts between the subunits which make the microtubule stiff and strong and difficult to bend. And they have a long persistence length, which means they can be very long without breaking or bending. So this is supposed to be the stiffest, straightest structural element found in most animal cells. Do you guys believe that? Bruce Albert says it. Yep. So that's good because you've got things that are going very long distances. If you think about neurons, right? And so you need to move these vesicles very long different distances. So you want something with a long persistence. So they have structural polarity. They've got a plus end and a minus end. And so does, okay, so actin monomers are monomers compared to tubulin dimers. And they assemble head to tail, and they also have polarity, a plus end and a minus end. And so when you're thinking about plus ends and minus ends, just think of the plus end of where things are most likely to add, and the minus end is where things are most likely to leave. Okay? So actin and tubulin filaments, they grow and they shrink depending on the concentration of free monomer in solution, which is a critical concentration. So this is just showing right here, this is your microtubule, and this is showing your, so remember, it's these 13 protofilaments that bind together and form your microtubule. And so here you can see they kind of play out as they're disassembling or assembling. So you have your individual polymers forming here. So association or dissociation is often greater at one end than the other. So one end can elongate faster or shrink faster than the other. So your plus end elongates faster and your minus end shrinks faster. Yes? So we're just getting a little confirmation here in terms of the concentration of monomer. When you're above your critical concentration, so when you have a high concentration of monomer, you're going to grow. And when you have a low concentration of monomer, you're going to disassemble. But that's in the absence of any of these proteins we're about to learn about that are going to completely mess with the critical concentration. Yeah.
Okay. So in this picture, is the plus end going to essentially just go like a squid and just snap together? Or
And
is
this-
this actually like unfolding and breaking apart?
Yep, so it depends on how you think about it. But here, so when you have, so the question is, in the plus end, are they going to come together or are they falling apart? If they're falling apart, then it's going to look like it's going to kind of play out.
Moving form was like being added, or just like, oh, it's breaking apart, so now they do. Because they're individual.
Yeah, so I think the way to think about it is it has to do with the length of each protofilament, and when you've got the 13 around, they're going to all interact and form that stable tube. Okay? Yes?
So, I don't know, but you were saying that the GTP bound, the cap, I guess, is really just the same active filament, right? It's just their GTP have like a different design.
Come again?
Like you were saying that those are the exact, they're no different than the other filaments, I guess, the actin monomers, but they just have like a different way they bind.
GT, are you talking about the tubulin or actin? You talk about tubulin and dimers
Yeah, and then that's what keeps it, or the
and
cap.
the GTP cap.
GTP.
Right, so the GTP cap, when you're GTP bound, you're more apt to add more.
But how does it
You're more
bind?
stable?
How does it know when to bind compared to the other? I guess. Like how does it know that's the end of it? Why wouldn't the GTP cap bind first before any of the other aspects?
So I think where we're lost is that you have to think of it as one, you've got all this monomer in solution, or dimer when we're talking about microtubules, okay? And it can't bind to the growing microtubule unless it's GTP bound, okay? If it's GTP bound, it cannot bind. And when it comes and it binds, GTP is hydrolyzed, but it takes a little while. And so as more and more add, because you're above your critical concentration, as more and more add, you're going to build up this GTP cap. And that's going to kind of protect the rest of the microtubule below where GTP is already hydrolyzed. Okay? But over time, as the concentration of monomer in solution goes down, there'll be less to add, and then the hydrolysis of GTP will catch up, and then you'll get removal. You'll get disassembly.
So it hydrolyzes from the top down.
You hydrolyze in the order that you're added. Okay? So if you add, right, so you're gonna add your dimer, right? And then you'll add your dimer. And so this has your GTP. And this has your GTP, right? And so since this one was added first, this will hydrolyze first, and then this will hydrolyze. But if your concentration is high, you're adding so rapidly that you build up this cap. But if your concentration is low, you're adding very slowly, and that allows the hydrolysis to catch up and you lose your cap. Does that make sense? Yes?
Is that an alpha beta dimer?
Yes, alpha beta tubulin. This is specific to microtubules. Yep. Okay. Yes?
It also said that in the dimer, it said there are two GTP molecules, but one cannot hydrolyze.
Right. So looking at this alpha beta tubulin dimer, the alpha tubulin does not have GTPase activity, cannot hydrolyze GTP. But it's the beta tubulin that hydrolyzes the GTP, and that's what we're talking about with the GTP cap. Okay? Great. Any other questions? No? Are we good? We have a few more minutes. Okay, so you could think of the plus end as the more dynamic end, because that's where things are more readily being added. Okay? So cells, they favor these energetically unfavorable processes to this spontaneous process of the growth and disassembly. So you can use it to push or pull an attached load. So like when we talk about, my favorite example is always chromosome segregation, because that's a really nice way, because you actually have attachment of proteins to your chromosomes, and then you have catastrophe or rapid disassembly of your microtubules in your mitotic spindle, and that's what pulls the chromosomes apart. Did I see a hand going up? No. Elongating actin filaments can help push out the leading edge of a motile cell. Yes?
This is just like a mechanical question. Is there some kind of binding test place when the active filaments hit the membrane?
Is there some kind of
Binding with the active filaments, pushing it to the membrane? Does it dilate the calcium flash? What keeps it pushing it to the membrane?
okay, so you're asking
Intermediary between the end of the active filament and the wall of the cell?
So you're asking about with active filaments when they're growing towards the membrane, push the membrane forward. push the cell forward, squeeze and back and film it going right through the plasma membrane. I think that the strength of the, well, let's see. I feel like the strength, the pressure would have to, the force would have to be so strong to get through the plasma membrane, and then you've got this hydrophobic. It's, I don't know how. I don't know. I don't know. Yeah, well, it does push, and that's how you have movement, though. But that's how you have the cell moving, is by kind of pushing up against the plasma membrane and causing it to protrude.
Maybe because it was so, the cell membrane is so fluid, it's not brittle enough to break through. Anytime it ever pushes against the membrane, it moves in that direction.
Okay, so Stephen is saying maybe the plasma membrane is so fluid, and it's not this brittle, it's not brittle, it is fluid, that maybe the actin is just pushing up against it and moving it forward. That's my understanding of how it works, but I'm sure there are accessory proteins involved. Yeah. Okay, we've got one more minute. I'm getting great questions today. You guys are keeping me on my toes. So this is what I was just saying. ATP, so with actin, you've got ATP bound, and with tubulin, you've got GTP bound, and ATP or GTP hydrolysis occurs when a subunit assembles into a polymer. So like I was just saying, you've got your monomer, and then you've got your, when you're added to your polymer, you're going to get GTP or GTP hydrolysis. It's part of being added. GTP hydrolysis or ATP hydrolysis reduces the binding affinity of the subunit to the neighboring subunits, making it more likely to dissociate, which is why when you lose your GTP cap, you're more likely to disassemble. Which I haven't even gotten to tell you is called catastrophe. It's my favorite word of the cytoskeleton. I think we're going to stop here today, and we'll continue at some point.